Hello, welcome to Bioclassical channel for NEET. So this is C4 cycle, which is also referred to as hatch and slack pathway. So where does this particular pathway of C4 cycle occurs? This occurs in plants which is present in dry tropical region, which has got very high intensity of light present and also which lives in very high temperature. So they have got some special type of anatomy which is termed as Kranz anatomy. So the meaning of the term Kranz is wreath. So this type of anatomy facilitates carbon dioxide fixation in photosynthesis. When you look at the type of cells, here this particular C4 cycle occurs in two places, one in mesophyll cells and the other in bundle sheath. When you look at mesophyll cells, carbon dioxide is fixed in mesophyll cells. And the carbon dioxide is accepted by phosphoenol pyruvate, which is a 3-carbon compound, to form a 4-carbon acid, which is oxaloacetic acid. This oxaloacetic acid is then transported into bundle sheath. This bundle sheath normally, if you see, are cells which are arranged around vascular bundle. So this C4 acid, which is oxaloacetic acid, in turn is transported into bundle sheath, and here in bundle sheath decarboxylation occur. Please understand the nature of bundle sheath as this region is impervious to gases. So carbon dioxide cannot directly reach bundle sheath here, and it should come only through mesophyll cells. When you look at bundle sheath here, the concentration of carbon dioxide would be really high, and the process of decarboxylation releases carbon dioxide, and in turn this carbon dioxide enters C3 cycle. And in C3 cycle, the major carbon dioxide fixation is enhanced by the presence of the enzyme called Rubisco, and the function of Rubisco would be on the higher side, as in bundle sheath the carbon dioxide concentration is more. So this carbon dioxide, which is released by C4 acid, would enter Calvin cycle, and thus the sugar is being prepared. And now after the preparation of sugar, the decarboxylation process, the C3 acid is getting formed, and that again would get regenerated and transported to mesophyll cells, forming phosphoenol pyruvate. The fixation of carbon dioxide in mesophyll cells is facilitated by the enzyme PEP carboxylase, which is phosphoenol pyruvate carboxylase. The major difference between Rubisco and phosphoenol pyruvate carboxylase is that this PEP carboxylase fixes carbon dioxide even at very low concentration, whereas Rubisco would fix carbon dioxide only at higher concentration. In mesophyll cells, PEP carboxylase is present, whereas Rubisco is absent. The same way in bundle sheath, Rubisco is present and PEP carboxylase is absent. Because of very high concentration of carbon dioxide, the efficiency of carbon dioxide fixation by Rubisco is enhanced, and hence the production. So by this hatch and slack pathway, C4 cycle, the production of biomass by photosynthesis would be on higher side compared to that of C3 plants. The example of C4 plants having such a peculiar anatomy, which is called Kranz anatomy, is seen in maize and sugarcane. So the examples are important for the NEET point of view. Thanks for watching. Subscribe the channel.